All right. Well, thank you all for coming today. My name is Kara Chizek. I am the instrument sales specialist with Akoya, and I'm here today with Dr. Avik Mukherjee, who's our senior technical applications scientist, and also with Dr. Surya Pandey, who's the immunotherapy assessment core manager at Northwestern University. And first of all, I want to thank Fadier for inviting us to come and talk with you about spatial biology. I know spatial is a hot topic nowadays, and when a lot of people think about spatial biology, they think about transcriptomics, so RNA based spatial biology, but we're using protein markers, and we're here to sort of talk about how to use protein spatial phenotyping as a complement to spatial transcriptomics, or just as a method on its own. And Avik's going to walk you through the PhenoCycler-Fusion 2.0, which is our newest high throughput, high multiplexing system for spatial phenotyping, show you some data and applications where it can be used. And then Surya is going to talk a little bit about how she's using our technology in her core lab and how you might be able to access the technology in her core. She has both a PhenoCycler-Fusion and a Vectra from Akoya, so she can do a wide range of protein spatial phenotyping in her lab. So, of course, if you have any questions, stop and talk to us after the seminar or ask questions during the talks. And just thank you again for coming.
Thank you, Kara. Thank you, everyone. I know it's a beautiful day, but we have got you stuck in a dark room. Today, I'm going to talk to you about how you can use Akoya's instrument and the imager and the chemistry to do spatial biology. The talk would be divided into three parts: why spatial, how you do spatial, and what spatial biology can give you or answer your questions. But first and foremost, when we say the word spatial biology, I hope these kind of images come to your mind, where you can look at hundreds of biomarkers, take any slide that you have and put it into an instrument and look into different biomarkers, look into tissues like tonsil, head and neck cancer, maybe looking into the prefrontal cortex of human brain. And in spatial biology, we have this arc of spatial biology, where how the researcher starts once they get an image. They do spatial phenotyping, then they go and try to look at if there are any rare cells that they have missed on their transcriptomic analysis, that now they can visually see exactly where located, and can be like a needle in a haystack situation. Look at functional state. Either it can be multi-omic, where you do a RNA-seq study, and then your sister section, you do a proteomic study, combine it, or you find, like, say, metabolic markers in a tissue and try to look at the functional states of the different tumors present in a single piece of tissue. You can do spatial neighborhoods. That means you look at a cell with different contexts and try to figure out how different niches are present in your tissue and how that can help you understand the disease. And once you find the spatial signatures, the clinical outcomes, then you can run maybe smaller panels and do high throughput study. And to do all of this, you need to do a whole slide imaging. Once you do the whole slide image, you don't have to leave out areas, and you can look into everything. And this imaging technology that we have in Akoya has been out there for more than 25 years, before even the term digital pathology was coined. And you use this technology to do this whole slide scanning of the tissue so that you are not leaving behind any cell and you can see the whole picture. Because there have been different technologies that give you a lot of quantitative data, sometimes qualitative, but they don't give you the context. For spatial phenotyping, you need the context. You don't need to take off the tissue or your cells from the slide, do an assay where you are not knowing from exactly where the cells came and what are the exact cells at the exact position located. And this kind of spatial phenotyping with proteomics have become more and more interesting because of the migration of nucleic acid-based technology moving into the spatial verse and joining with proteins to maximize your information from a small, precious piece of tissue. So why spatial biology? To address that, this is a meta-analysis that was done and published in JAMA Oncology 3-4 years back, where they were looking at the ideal predictive power of an assay. They were looking at immune checkpoint inhibitors, and they were looking at the receiver operator curve that you see on the y-axis, you can see it had to be an ideal biomarker. It needs to have more than 80% predictive power. When you look at the single marker immune checkpoint therapies that were available during that time, or even looking at tumor mutational burden, you see they don't give you the diagnostic accuracy as a protein spatial phenotyping. And a very fun thing to mention here is that when this meta-analysis was done, they looked into papers with only 3 protein markers. That means with even 3 protein markers, you can get a better understanding of immune checkpoint therapy or even stratify patients into right therapy group than other single marker RNA expression or NGS technologies. And this can be seen with this cartoon, because when you look at this cartoon, you know that no cell in a tissue is an island. It's important to know what cells are present, and more important to know where they are present and where they are present in your tissue respective to each other. And that can determine what they are contributing to the biology of your tissue. For example, if you look at these 2 tissues, you grind them up and look at the tumor infiltrating scores, they don't look too much difference, as you can see here. But if you look at these 2 images, there is a difference between the image on the left and the image on the right. And that's the power of spatial biology. When you image with protein markers, you can really better understand what's exactly happening in your tissue. There was another study done where they were looking at survival of patients for PD-1, PD-L1 checkpoint therapy. What they saw that the patients that responded to immune therapy had less amount of immune cells in their tissue, while the patients that didn't respond to immune therapy had a lot of immune cells in their tissue. This kind of sounds counterintuitive. Then they went down and they took a cytotoxic T cell and drew a 30 micron radius around it. And they saw when there are more Tregs present in that 30 micron radius, those cells were suppressed, and those were the tissue types of non-responders. And when there was no Tregs present near the T cells, there was no suppression, and those patient tissues were more responsive to immune therapy. So not only knowing how many cells, where they are, exactly precision location of them, and the physical interaction between different cell types can determine your disease state. And that kind of gives you this whole arc of spatial biology from spatial phenotyping to signatures. Another interesting case study where this group, they have been looking at breast tissue. This is a stage 2 breast cancer tissue where they used 36-plex panel. In this image, there were 63,000 cells. They looked at different 12 phenotypes, but they got 3 distinct population here. If you see in this Disney plot, there is this small red island, and there is a very small orange overhang. And this orange overhang is like small number of cells, which are luminal epithelial cells. When they have done like a single cell RNA-seq, they have overlooked thinking that that's an artifact. But here they can exactly see where these cells are located or where they're coming from. They identified these 44 luminal cells. They looked back into their RNA-seq data to corroborate that they found these cells there, and they saw they found it, but they ignored it. Now they know where they're located, and they have stem-like properties. So now they're doing even deeper studies to understand whether these kind of rare cells can be found out, and can they have an effect in early detection of breast cancer. The next study that I'm going to show you, if you go to Akoya's website, you will see this image a lot. This is an image from a patient who was HPV plus, but partially responded to immune checkpoint therapy. The patient passed away after 14 months. But prior to that, the group had the pre-IO specimen so that the group can ask the question why there was a heterogeneity in the response in the patient, why there was a partial response. And I always feel when you look at the H&E image, where you know this is the tumor region, when you do immunofluorescence, I think it doesn't add complexity, it more adds like a value to what you can get more out of that single piece of tissue. So what they did, now we are going to look at the same piece of tissue with different markers. You can see CD3 is dotted throughout the tumor microenvironment, telling us that some biology is happening, but it is not identifying all our B cells. So we add one more layer of B cells. We see the B cells are also dotted through the tumor. We see the germinal centers in dark blue, but still we don't know where our cells are relative to the tumor. Then you add some more things in the protein level to see there is some infiltration of macrophages and dendritic cells up here, but not down here. If you want to know what subtype differentiate between the subtype of macrophages, you add few more. Now you can see the checkpoint expression in the microenvironment, but we are lacking the tumor context. Then we add few more. So basically, I'm trying to show you're looking at the same piece of tissue, adding more. Now you look at the structural data, vimentin, SMA1. You can see CD31. It looks like a highway through the tumor. And using this panel, it's telling us that ultimate understanding of the tumor microenvironment can be done. Now you can see there are 4 tumor regions. Now you want to know what are the functional state of the cells. So you add the metabolic markers. Once you add the metabolic markers, now you can see that the bottom part has increased expression of GLUK1 G6PD expression, high glucose uptake at the bottom tumor, and none of the red signal is seen in the top. This can give you a clue that this can be one of the reasons why there was a partial response, because the tumor cells are operating in different metabolic states. Now you want to see stress data. So when you put the markers, you can see a lot of pro-apoptotic signals seen up here compared to the tumor here. This kind of gives you the idea that there is a heterogeneity inside the tissue between all 4 types of. So finally, what happened is we took all these panels, it was 101 proteins, they looked into the tissue and they got this 101 biomarkers, and they had different kinds of panels that they created. You can look in the bottom left. And this is the first of a kind where they published 101 biomarkers in Gen Biotechnology. And they were able to look at how there is the intra-tissue heterogeneity. If you look at the top, you can see that there is a tertiary lymphoid structure invading into the tumor area where there is no such structure in the bottom. That's the idea of how you can use as many markers you want, up to 101 plex. You can image it, stain it, and you can analyze it to understand. When they did the analysis, what they looked at all 4 tumor regions. They were able to look at tumor 1, 2, 3. They were able to look at the tertiary lymphoid structure. They are able to look at the germinal center in the normal tonsil. They have to look at the tumor 3 where there is no such invasion of a tertiary lymphoid structure. They can look at the ducts, esophagus, and different squamous epithelium that is present. Next, what they wanted to do after this kind of correlation matrix that they got from different markers, they wanted to compare the functional state with the protein markers, the immune markers, and the metabolic markers. And as you can see, okay, so what they did is they looked at the tertiary lymphoid structures up there, and they understood that the difference in the metabolic state of the tumor were able to help them understand the intratumoral heterogeneity. So not only you can see intertumoral heterogeneity, you can see the intratumoral heterogeneity here. So they went through the things where they looked at the spatial phenotyping, they found distinct neighborhoods, they were able to look at the functional state, and finally, they were able to look at the metabolic profiles. And all this is possible because of molecular barcoding, where we use the chemistry, which I'll talk more details a little bit later, using barcodes, not a primary, secondary antibody. So that helps you to scale up from tens of antibodies to hundreds of antibodies and look into the complexity, because our system doesn't use any harsh method that can damage your tissue. It's more isothermal. So after they got this TLS, they wanted to see what is the difference between the tertiary lymphoid structure and the germinal center. So they did a region of interest-based transcriptomic studies where they picked up the germinal center from the tissue and also the TLS, and they looked at what were the RNA transcripts present there. We have just partnered with Thermo Fisher to utilize their View RNA product with our PhenoCycler-Fusion. So they created these two antibody panels for lymphocyte activation and immune activation. Along with they had some control panels. What they saw is that in tumor 3, there was no upregulation of CXCL9 or CXCL13, which are the lymphocyte activation and recruitment chemokines and cytokines. And also they saw that the immune activation and response panel was seen in the tumor 4, which was invaded by the TLS, but not in tumor 3, where there was no invasion. And when they put it together with the protein data, they were able to understand that you can catch the chatter between the cells, all the diffuse chemokines and cytokines with this kind of a small RNA panel. And you can understand that the ones where there was no infiltration was due to the expression of MMP9, which can be looked at the tumor invasive margin, whereas CXCL9 and CXCL13 were recruiting the CD8 T cell and B cell to kill the tumor. Now, we looked at a case study which looked at cancer, but there are other applications. People have used PhenoCycler-Fusion to look at Alzheimer's disease. You can see the panel for looking at immune cells, astrocytes, oligodendrocytes, neurons, and the Alzheimer pathology. They were able to image the barriers, blood-brain barrier, meninges, and also the accurate pathology of Alzheimer's disease. People are also using mouse tissue with PhenoCycler-Fusion. This is an example of a paper that got published where they looked at lymphoma, breast cancer, and melanoma using mouse tissue. People are creating neuromouse panel, customizing their own panel. And this is a picture. I always love to show beautiful pictures. There's a beautiful picture of a neuromouse FFPE. So why I'm showing you different tissues and different things, because for a spatial proteomic platform to be acceptable and being worked on with different types of tissue, it should be highly deployable. I mentioned the term Codex up there because that was the old name of the technology. When the technology, if you go and look at papers, it would be called Codex. You can see different kinds of tissue. I don't know whether you're interested in colon or thyroid or kidney, renal cortex, oral cancer, placenta. So you can see there are different kidney cancer and even like mesothelioma. So PhenoCycler is a widely deployable technology and you can use it. Now the question comes, how do you do that? Well, these are the technologies. In Akoya, we have this continuum from discovery to translational, where if you need high-plex, you use PhenoCycler-Fusion, where PhenoCycler is this box and PhenoImager-Fusion is this imager. Or if you want to go high throughput, you can use PhenoImager HD, where you can go up to whole slide scanning of 7-plex. That means 6 markers and DAPI, and you can do 300 slides per week. Whereas here you can look at hundreds of markers and do a throughput of 20 plus slides samples per week. So with PhenoCycler-Fusion, you get the advantage of both, where you can do 100 plus biomarker, also 100 plus slides per week. And we are going to talk how you do that. This is PhenoCycler-Fusion 2.0. When the 1.0 was launched, you can use only 1 slide. Now you can do 2 slides at a time. That means when 1 slide will be in imaging, other slide will be in the fluidics. These are some of the features. The imaging area, as you can see, is 35 by 18 millimeter. You have a throughput of 20 samples per week. I'll show you the thought experiment, how we have calculated this data. You can run 2 distinct panels in 2 separate slides in a single run. Or you can run 2 different tissue types, but same panel in the same run. And the good thing is the data side is in gigabytes. So you don't have to worry too much of terabytes of data and how you compress it down. The basic chemistry is you have a primary antibody with an oligonucleotide barcode, and there are reporters, which are antisense barcodes with fluorophores connected to it. During each imaging cycle, 3 of these reporters go and bind to their complementary barcodes, and you image it. And that's what gives you the signal. The step 1 in PhenoCycler-Fusion is you take standard histology slides and fresh frozen or FFPE samples, and you take all your primary antibodies and put it onto the tissue. This doesn't mean if you have a FFP sample, you are not doing all the way you treat a FFP tissue. You are going to do your deparaffinization, dewaxing, and everything. But for antibody addition, you take all your primary antibodies and you add it to it. Then we provide you with a flow cell, which goes on top of any normal 1/3 histology slides. And this is the input through these holes. The fluid is going to flow. And this is the imaging area that you have for this study. And then this slide goes on to the PhenoCycler specific slide carrier, which goes into the imager. And when you click run, now the process is automated. What happens is the PhenoCycler will send in 3 reporters. And DAPI, the reporters will find their protein counterpart and they will hybridize and get settled. And the DAPI will light up your nucleus. And then the imager will do the automatic imaging. After the imaging is complete, the PhenoCycler will send in buffer that will dehybridize the probes. And your tissue goes back to the phase where it has all your antibodies and is ready for the next round of reveal, image, remove. So basically, you are not doing any stripping, all isothermal reaction, because basically you are dehybridizing the markers and you have all your protein markers there. So as it's connected to a barcode, you don't have to worry about the species of your primary antibody when you are using this assay. So there is no host-to-host cross-reactivity. With 2.0, as I said, you can learn a single large tissue. You can do TMA. Our imager has a TMA specific algorithm which recognizes each core and scans them as TMA. You can do 2 different types of tissue in the same slide. So whenever you look at the cost of running a sample, it's per slide because of this huge area. You can run multiple samples on the same slide, or you can run huge chunks of tissues. You can run 2 panels for 2 different slides, or you can run 1 panel for 2 different slides based on your need. And this is the thought experiment to show you how you can do 20+ samples a week, where you can start on a Monday, do all your staining. It's a 6 to 8 hours protocol. You batch stain all your slides, put 2 of them in PhenoCycler and go. And next morning you have the image and you put 2 more slides, and that's how you get the imaging done, 20+ samples per week. This is for if you are doing 50-plex. If you are doing 100-plex, it would be a little bit different. And as I said, we do molecular barcoding. These are some of the standard chemistry that people use with the protein antibody with a secondary with HRP or a secondary attached to a fluorophore. But we are doing molecular barcoding where it gives you this huge scalability to go from 10, 20 to hundreds of markers. And when you look at our antibody panels, you can select the antibody clone that best works for you, or you can get an inventoried antibody. That means which is already attached to a barcode and you can buy it as a complete assay from us. You can get a list of screened antibodies where we have screened the antibodies for barcode compatibility and we can give you that information. And there is community screened antibodies where our customers who have published their data, you can get the clone information and barcode compatibility from them. We right now have 80+ unique barcode. That means if you have your antibodies that work in your assay and you can put 80+ barcodes and do 80-plex right now. These are some of the examples of the immune markers that you can look in human FFP samples. And all this information is available on our website, and I will provide Fateh with the PDF of this slideshow. We can look back later. These are some of the examples of neuroantibody. And we also showed mouse data. So there is some mouse FFP antibodies that are available right now. You can buy them singularly. There is not like a kit, but you can buy them a la carte and look into your mouse tissue. So you know how the staining is done, how the imaging is done. The next thing is, the most important thing is image analysis. So the image that comes out of the machine is a QP-TIFF image. It's a pyramidal TIFF image, which can be analyzed with any of these software systems. We have created a open software system using QPath and Cytomap. In QPath, you do cell segmentation, and in Cytomap, you do the spatial analysis. You can, if you have access to something like a Visiopharm or Indica Labs Halo, you can do your analysis using them. You can use CURAT. There is a ScanP port that has been programmed, that has been R-based program launched in CURAT that you can use. So you do the cell segmentation in QPath, then you move to do the spatial analysis in CURAT. We have also partnered with Enable Medicine. That's a cloud-based image analysis software where you can do image analysis. So there are plenty of options. And if you have a bioinformatics department who are great at writing Python codes and packages, you can get your custom codes to work on these images. There is no issue. This is basically an image analysis pipeline where you do data processing, cell segmentation, quality control, clustering, phenotyping, and spatial analysis. The first step is you take your multiplex image and divide it into single cells. That's the cell segmentation step. You can use an algorithm like StarDist or a deep cell algorithm to do that. After you have done that, you need to make a dimensional reductional plot, like a UMAP or a t-SNE plot based on which kind of program you're looking at. First, you get single marker plots, and then you can annotate him. You may have looked into t-SNEs and a lot of UMAPs while doing single cell RNA-seq. The great thing here is you have the image to go with it, so that when you do the annotation, you can see whether you are annotating and picking up the right cell based on the single marker expression. Then from this, you can create like a correlation matrix, as I showed you earlier. If you have proliferation marker, epithelia marker, vasculature, T cells, B cells. After you get all these data, you can do a spatial proximity analysis to look at the distance, the temporal profiles of the cell types. And then you do the neighborhood analysis. It's a Python script that can be downloaded from GitHub and can be worked on, which look at how there are different niches. And presence and absence of these niches can help you understand how you're looking at your responder versus non-responder. So what spatial biology can do, I'll show you some of the published data. This is an example where this group was looking at colorectal cancer, and they had two different patient types, the D2 and CLR. And as you can look at the Voronoi diagrams in the bottom and the top, the staining, you can see there is more organized structures in the D2, in the CLR patients compared to the D2 patients. So when they did a neighborhood analysis, and this term neighborhood can be very loosely depicted by this cartoon, where if you think of a Chicago metropolitan area, you have the suburbs, you have the city, you have the rural areas, they all combine together, and there are roads leading to each area. And they make up the whole metropolitan area of a city. So when you look at a tissue, they also have different kind of cellular niches. In this case, when they were looking into D2 and CLR, they found the neighborhood 5, the follicles were absent in the D2, and that's why they were not responding to immune therapy that were applied to those patients, whereas the CLR were more immune responsive because of the follicle and more organized superstructures present. They were attracting T cells. This kind of similar study has been done in looking at the tumor heterogeneity in high-grade ovarian cancer, where you can see they looked in 23 tissue types and classified 19 cell types. And when they did analysis, they were able to look at the difference between the primary and the recurrent tumor types. Now more and more studies are going not only proteomics, but adding proteomics with single-cell RNA-seq. This is an example where they were mapping the cellular biogeography of the human bone marrow niches with single-cell RNA-seq in a sister section or a serial section, and PhenoCycler-Fusion on the other serial section. These are you can see the different antibody panels and how they did the proximity analysis and did the violin plots to look at the expression and the functional states of the cells. This is another study looking at a multi-omic atlas. So people are doing a lot of this hub map and human cell atlas kind of studies where they are creating these atlases to look at the tissue and figure out how the RNA and the protein look together. This is another study which is part of HubMap. They have also created different softwares where you can take a multimodal data, one coming from your single-cell RNA-seq, one coming from your proteomic data from PhenoCycler, using something like a MaxFuse, which takes in this kind of cross-modality data, and they can do a analysis to overlay your UMAPs and your t-SNEs and give you the accessibility of a functional state of the cells. There have been data published from Jackson Lab, where they were doing the xenium, 10x xenium, and then they followed it up with PhenoCycler-Fusion on the same slide. So this can be done with any other kind of RNA assay where you can do targeted RNA, then targeted protein on the same piece of tissue, because the whole thing of multi-omic is, and also they did H&E staining on the PhenoCycler, because after every PhenoCycler run, you can take off the flow cell and do H&E. So basic goal is, this is just a conceptual diagram that you want to know your RNA, you want to know your protein, go into that single cell and come back and understand the whole picture of your tissues. When you look at this kind of high-plex study, this is a 56-plex study that was done to understand the cellular neighborhood of responders and non-responders. They found a spatial signature. They found this signature, which then they can create a 7-plex panel, and then you can use just the PhenoImager Fusion to run that through multiple slides, and they can get a spatial score looking at what is the distance of the T cell from the tumor or from the Treg. So the instrument PhenoCycler-Fusion can work as a high-plex when you are using it in combination with the Codex box or the PhenoCycler box, or alone, it can take in 4 slides, which can image up to 7 colors and give you a high throughput data. So you can go between hundreds of markers, hundreds of slides. In the end, I will just leave you with the question that you can see this data, beautiful pictures, quantitative data, significant discoveries. What you will want to do depends on you. I hope I have been able to rekindle some questions in your mind, how to use this tool in your future research. And if you have any questions, I'll be happy to answer. Thank you.
So you mentioned that this 10x study was done on the same slide, or you
same slide. So yeah, this is Jackson Labs. They have put this protocol on protocol.io, where they took the xenium, as you can do it with the normal slide. So they did the xenium study on the same tissue. They came to PhenoCycler-Fusion and ran a protein panel, and that's the image that they put in for both. It's the same tissue. Xenium first.
But like the same slide or the same tissue?
Same slide, same tissue.
And xenium.
Yeah, xenium first. People have been doing, this is off level, like also same with Cosmics. Cosmics first, then PhenoCycler. Yeah, you can see the whole protocol.io is, they have given step by step everything that they did. And they can do H&E. So with every PhenoCycler run, after you have done your IF, you can take off the flow cell and you can do H&E.
Hello, everyone. Thank you for inviting Fatih and Kara and Avik. Good to be back here, obviously. So my name is Surya Pandey, and I run this immunotherapy assessment core at the Lurie Cancer Center at Northwestern. And today I'm going to tell you more about our core and how we use PhenoCycler-Fusion there and how you can access it. So just to give you an overview, we were established in 2019, like late 2019, and we are located in the downtown campus of Northwestern, 4th floor of Terry Building. Our mission is to provide basically one-stop solution for all of the immune monitoring techniques for dissecting mechanisms of immunopathogenesis and exploring novel disease markers. We have full service capabilities, meaning you can just drop off your samples and get your data. We can take the project from start to finish, and we provide both self and staff-assisted services. So we will train you on the technology, data analysis, and everything that we run in the core. So just to tell you more about the major services that we provide, we have a GeoMix DSP for spatial transcriptomics and proteomics. We recently got Immunospot analyzer that can do ELISpot enzymatic assays, and also up to 4 colors Immunospot like fluorospot assays. We have a Cytek Aurora for 24 colors spectral flow cytometry if you need to do deep immunophenotyping of your cells in PBMCs or in tumor single cell suspension. We do immunostaining also for those samples if you need us to do. And then we have automated stainer from Leica Bond that can do automated immunohistochemistry. We have 10x Chromium for single-cell RNA-seq. We have a multiplex ELISA platform from Mesoscale Discovery, where you could go from 1 to 20-plex for your sensitive ELISA detection needs. Then we have other small peripheral equipments and also provide tissue dissociation services in addition to consultation and data analysis. You can go to our website here, and we have 60 service lines. So if you go there, you can find everything in details. Our turnaround time is ranging 15 minutes to 3 days, depending upon the technology. We also have a capability to manage larger number of samples. We do provide some technician time to develop custom protocols if you need them. And then all of our services can be customized according to user-specific needs. Avik has told you, I think, everything about the PhenoCycler-Fusion platform, but I will tell you from my experience and how we have used the technology in the core. So first question that I tell the users to think about is between both the low-plex platform and high-plex platform, how would you decide? So it depends on your research goals. Do you need really like very high-plex markers for your study? Or you can do with like 3 markers, 4 markers, 6 markers, and be done. So that's why you really need to think about whether you need a low-plex technology or a high-plex, because depending upon that, your time, your money, and resources will be different. You can think about throughput, whether you want to do 1 slide, 2 slides, 5 slides, or you have a big cohort of patient slides, like 20, 30, 100. So that will also decide which platform you want to choose. Antibody availability, of course, when you're doing higher plex, you're depending upon the barcoded antibodies. And although they are available for most of the markers, all the time researchers, they want to use their novel antibodies of interest. And sometimes they won't be available as barcoded. You need to conjugate them, or it will take more time. So if you don't have that time and you really want to use your study to get the data fast, you can go for the lower plex because it doesn't need barcoded antibodies. And again, the tissue embedding. Mouse studies, they are mostly done with OCT embedding frozen sections, majority of times. Some people do FFPE also, whereas human samples, they are usually FFPE embedded. And the lower plex platform, I would say, is more suited for FFPE embedded sections. So keeping all these things in mind, you can decide which way you want to go. We have been using the lower plex platform that is Opal-based workflow in our core for a long time, like last 3.5 years. So I will tell you more about those and the examples of those, because Avik has told you all the examples about the higher plex platform, and the data that we have on the higher plex, like Codex-based studies, is not published yet, so I can't show you that. So yeah, for the lower plex platform, the way we do it is we design the panel first, then we go for panel optimization by pairing your antibodies with like proper Opal dyes. Then that we do in our automated stainer for immunohistochemistry from Leica. Once the staining is done, then we image it by this microscope, like the imaging part of PhenoCycler-Fusion. This we have done for a lot of projects. So we have validated immune marker antibodies and panels. We have done in a lot of human tissue sections like brain, skin, muscle, ovarian cancer, heart, bone marrow, different types of tumors. And we have done some on mouse tissue section also, like tumor and spleen. What we have found is the technology is really robust. It gives you really good quantitative data. We do provide you with all the panel design support and the data analysis support also. So this is the first example that I want to show you from the lowerplex platform. This is a 6-marker panel that was developed in the core, and this is not Opal-based. Sorry to tell, this was like a fluorescent conjugated antibodies, but we developed this for mouse frozen sections for a mouse spleen, and you can see we have stained it for CD3, CD4, CD8, CD19, and Ki67 for proliferation. But this is just to show you how it can be adapted for multiplex like fluorescent imaging techniques and not necessarily Opal dyes. But Opal dyes and therapeutic signal amplification has its own advantages, so we can talk about it if you want to. The second example that I want to show you is from Sonabend Lab at Northwestern. They work on glioblastomas. So this is a paper they published in 2021 where they showed that ARC1-2 phosphorylation predicts survival following checkpoint blockade immunotherapy in recurrent glioblastoma. So what they did was they stained their glioblastoma tissues from 2 patient cohorts with 6 markers. Those were like SOX2 for tumor cell marker, TMEM 119 for microglial marker, CD163 for macrophages, ADC. And you can see here they did like H&E staining and PERK staining. So previously they had shown that BRAF and PTPN11 mutations enriched MAPK and ERK signaling pathways. So they were highly motivated to look into the mechanism of that. And that's why they started looking into phosphorylated ARC expression in these cells and how it was contributing in better immune response to this checkpoint blockade in the tumor microenvironment. So once they stained the slides with their panel, what they found out was the PRC positive cells were more expressed in SOX2 positive cells, like in the tumor cells, as compared to the other myeloid cells like TMEM 119 and CD163. And similarly, they compared the SOX2 positive and PRC positive double expression in high PRC glioblastomas and low PRC glioblastomas. And they found that this double positive population was higher in the higher phosphorylated ARC population. So they extended this study further and did a spatial analysis to see the interaction between the SOX2 positive and PRC positive cells and glioma infiltrating myeloid cells. So what they did was they calculated distances between the SOX2 cells, like the tumor cells, and the myeloid cells that were TMEM 109 and CD163. And what they found out was the distance from the TMEM cells was less for the higher phospho-ARC glioblastomas as compared to the lower phospho-ARC, meaning in this tumor microenvironment where there was more phosphorylated ARC, the tumor cells were interacting with more immune cells or myeloid cells, and they had higher expression of MHC. And similar for CD163 also, in the higher phosphorylated ARC environment of glioblastoma, the distance was shorter between the tumor cells and immune cell types. Taking together this data and other single cell RNA-seq data and other technologies that they have, they reached to this conclusion that they can predict the survival of a subset of population of recurring glioblastoma patients following an anti-PD-1 immunotherapy. So this lower-plex platform has different moving parts. So if you need a project consultation for this, we can talk about it. And because it requires different kind of optimizations and has a different timeline than your higher-plex Codex platform. So as I said, I don't have data for this PCF or the Codex platform, but I can tell you the cost breakdown so that you know, because this is the first question I get from anyone who wants to run a PhenoCycler-Fusion or Codex. So for a mouse frozen 12-plex section, the cost would be around $1,600 for the service only. The users need to buy their own antibodies. The rest of it, we provide the reagents and runtime and all the 3-day protocol that needs to be done for this workflow. And for a human FFPE section, the cost would be $1,900 per slide. Here are your advantages, why you should do it in a core rather than doing it in your lab. It's just more cost savings and you get more expert resource for staining, imaging, data analysis, and troubleshooting. Here is a panel that we have bought for all the PCF antibodies that you can buy from the core, and so that your cost for antibody will go down and you can add your antibody of interest in the panel and run, say, a 10-plex, 12-plex, or 15-plex, or even a 25-plex panel. Avik has already showed you the area where you can put your tissues, so I would highly recommend you to think about this also. You could fit a lot of tissue sections. I have seen people putting like 40 tumor, like 2 millimeter punch biopsies here for tumor microarrays, and even 6 mouse hearts. So just to give you an idea. And finally, this is all the information, how you can get in touch with me and ask for project consultation. And if you don't see it here, you can always ask Fadi. He knows how to reach me. If you have any questions, I'll take them.
I'm wondering what kind of data analysis options do you offer for, especially for the
So the low-plex, we have Akoya's Inform. That is a machine learning-based software. We have license for that. And the higher plex, we have just bought the Enable Medicine license, and we also do QPath in addition to that.